going on. So thanks everyone for joining us today on a Saturday morning for today's webinar 18. And before I introduce our today's speaker and we start the today's presentation, I'll just briefly take a few minutes of our time and introduce our center and why we have been doing these webinars for the past almost 2 years now. So yeah, I'll just take a few minutes. Okay. I'll just request everyone to please keep themselves muted and also during the course of the presentation. And if there are any questions even during the presentation, please only type in the chat box, but please do keep yourself muted. So Kadambari, can you make sure that
Yeah, sure.
And if there are people who are joining, please do admit them. So, yeah. Okay, so I'm Surat and I'm a senior research associate at the Center for Predictive Human Model Systems at Atal Incubation Centre, CCMB. And this was a center that was jointly initiated between Atal Incubation Centre, CCMB, and Humane Society International India. And we are a science and policy center that has been now it's almost 3 years since we have we started. And the idea of the center is how, as we all know, that biological systems are predominantly animal models are used in various kinds of biological research. But in the last few decades, several other model systems have been developed, which have greater relevance to human biology because they are based on human cells or human biology. And yeah. So these are also referred to as human relevant model systems, for example, organoids, organ-on-chip, and also 3D engineered tissues such as an ex vivo cultures, etc. And many of these model systems can generate human relevant data. And using this human relevant data, we can now build in silico models, which can capture and predict various subtleties of human biology, which very often animal models fail. So towards this end, our center has been working towards various initiatives in terms of how we can further enable this kind of science in India. And towards this, our work has been divided into various subgroups. So one branch has been how we can further increase awareness for these methodologies, because while this field has been growing at an exponential rate globally, in India, the awareness for these methodologies is still low. So we have written several popular science articles, reviews, and this monthly webinar series is again an initiative towards that end. Similarly, we have also conducted several education and training workshops, and we will also be conducting the first ever India EMBO lecture series in this particular field of microphysiological systems. And I'll be talking about more in one slide down. And you've also been involved in several policy initiatives where we have written 3 white papers, and we have conducted 2 multi-stakeholder roundtable meetings with government and regulatory bodies to understand how we can have more policy initiatives to advance this field in India. And we are also involved in several global collaborations to increase the knowledge base of this field, both in India and globally. And we also provided grants to 2 Indian scientists to develop an adverse outcome pathway in the field of cancer. And both of these adverse outcome pathways have now been submitted, and we'll also come up with more updates in this area soon. So the way we envision the roadmap to enable a more human relevant paradigm in India is one has been, we are working towards establishing dedicated centers of excellence to further validate and develop these methods. Another aspect is how we are trying to enable connections between the technology developers and academia and the end users and pharma companies and others, and so that the technology transfer process can be a smoother this thing. And we also engage with various regulatory bodies during the initial stages of technology development itself, so that once the technology is developed, so the process of regulatory approvals is also a much faster and a smoother process. And as I've already said, that in many cases, the global scenario for these technologies is much more advanced. So another aspect that we are working on is how we can further enable cross-border collaboration for both technology transfer and to get more international funding. And specifically in India, we are also trying how we can indigenously develop these technologies, and towards this, how we can enable various supply chain issues that we are currently having, which can both reduce the cost and again increase the development of these technologies in India. So with this, I would just briefly talk about some of our upcoming initiatives. So one is, as we have, as I already said, that the Center for Predictive Human Model Systems, along with CSIR-CCMB, we are organizing an EMBO India lecture course in the month of October, 31st October to 4th November. So the registrations for this lecture course is now open. So please log on to the link given here and do register for this lecture course if you are interested in getting to know more about various advances in organoids and organ-on-chip technologies. And we have speakers from across the globe who will be coming and taking these lecture courses. Secondly, we also have a quarterly newsletter. So where we have various news articles, informational videos, and opinion pieces, which again talk about what is all the science which is going on in this field. And we'll put the link in the chat box for the same. And third thing is we're also developing an interactive database of various microphysiological systems researchers in India. And if you are a researcher, an independent researcher working on organoids, organ-on-chip, or a computational methodology in India, do fill this Google form so that we can keep updating this interactive database, which again will be launching very soon. It will be an open access interactive database where anyone can kind of see who is a researcher in India who are working on various human relevant technologies. So this is another initiative. So please fill this form if you are an independent researcher working in this field. So with this, I will stop, and I will introduce our today's speaker for the day. So Dr. Ganesh Bagler, who is today's speaker. He did his PhD in computational biology from CSIR-CCMB, and then he did his postdoctoral training from NCBS Bangalore and Max Planck Institute for Molecular Genetics, Berlin. And his lab is currently exploring the systems architecture of complex diseases, drug target networks, and brain networks. And broadly, the areas that they study include computational and systems biology, bioinformatics, mathematical modeling, network biology, in silico drug discovery, and biomedical data analytics. So we are pleased to have him here for our monthly webinar series. And with this, I will over to you, Dr. Bagler, and thanks a lot for agreeing to speak at our webinar series. And I would request all our listeners to, if you have any questions, please type in the chat box, and we'll take all the questions towards the end. Thanks a lot.
So thank you. At the outset, I must thank the Center for Predictive Human Model Systems for inviting me, and particularly Dr. Surath for having me around here. It's my pleasure to be coming on this forum of AIC-CCMB and Humane Society International, and talking about network biology, research that we have been doing in my lab, which is primarily inspired by graph theoretical modeling of biological systems. And that's what I'll be presenting today in this around 40 minutes presentation. And I would try to keep it at a level which is accessible for undergraduate, postgraduates, and also PhD students who are coming from wider disciplines, so that while I'm inspiring them to use network modeling or graph theoretical paradigm for understanding biological systems, that's what my intention is. Right, so I am from Center for Computational Biology, IIIT Delhi, and I've been here for a while now. Our center, as well as I am affiliated to Infosys Center for Artificial Intelligence, our agenda is to look at biological systems from a complex systems perspective. And I'll explain a little bit about what are these complex systems that we are referring to as I go along. A little bit about myself. I've been trained in the areas of physics, astrophysics, followed by computer science, after which I joined CSIR-CCMB for my PhD. So I am an outlier, if you may say, in a place like CCMB, who didn't have any background in biology and had joined CCMB to do work on the interface of physics, computation, and biology. And it was at CCMB, while I was doing my PhD, that I started using graph theoretical models for understanding protein structures, about which I'll be detailing a little bit as I go along. And after which I have taken a long route to move from my initial aspiration of being an astronomer, from astronomy to all the way up to gastronomy. Today, our lab is considered to be the ground zero of what is newly coined, newly created niche called computational gastronomy, which is not the topic of today's talk. Nonetheless, I'll be touching upon it as I go along later on. My lab, the Complex Systems Laboratory, has been at the forefront of these efforts on modeling biological entities, biological systems from a graph theoretical perspective. So to begin with, what are these complex systems or biological complex systems that I'm referring to? Any system which comprises of large number of sophisticated functional elements which are subtly interconnected with each other so as to perform specific tasks that otherwise cannot be executed by subset of the systems are referred to as complex systems. And I know it's a mouthful of a definition. However, in a short summary, I can say that all of these systems can be characterized with this phrase that the sum of parts is not the same as the whole. Even if you understand the behavior, let's say physiology of individual elements, when they are put together, something else arises out of nowhere. Emergent properties come out in the complex systems, which are not anticipated by simply looking at its individual elements. So whether it is protein structure, complex diseases, or brain networks, or for that matter, food, all of these can be seen from the lens of complex systems and can be understood in a better manner by applying various mathematical and computational paradigms, of which specifically I'll be touching upon the graph theoretical paradigm. In my lab, we have been looking at biological systems from the perspective of modeling them, further controlling them, finding ways of controlling them, and also getting inspiration from biological systems thanks to their robustness and adaptability for designing engineering systems which can be inspired by biological entities. That's what we have been trying to do for a while now. The talk that I'm delivering is divided into three broad topics. To begin with, I'm going to tell you a story which hopefully will inspire you in looking at biological entities from an integrative perspective as a networked entities. Further, I'll be giving you an introduction to the network biology, which is essentially graph theoretical modeling of biological entities, biological systems. And while doing so, the agenda here is that to keep it at a level which is accessible to you, and the rest of it, which is a little more technical and something which is probably is not going to cut across all the audiences today. I'm going to leave it in the slide. I'm going to pass through them and we'll leave it to you. I'll be passing the slides to the organizers so that you can, if you wish, you can go through them later on and deep dwell on it as time permits to you. And finally, in the last part, I'll be talking about some of the applications of network biology, different domains, different aspects of biology where network modeling can be, or graph theoretical modeling can be used for understanding them better, is what I'm going to touch upon towards the end. So to begin with, most of you, I presume, are from the biology background and would be absolutely aware of the central dogma of molecular biology, which talks about information transfer from DNA to mRNA to all the way up to protein, which folds into a three-dimensional structure, giving its functional form eventually. Well, this has been there for a while. So can we understand biological entities purely from this central dogma, or do we need to think of other ways of understanding biological systems. And towards that, I'll be telling you a story of a biologist who finds a functional radio on an island. The idea here is to tell you that, look, if you find something which is absolutely functional and if you don't understand it, how would a typical biologist would look at it? And that approach is called as reductionist approach. And a radio such as this, if you were to understand its functionality from a reductionist paradigm, then what a biologist would do is to buy or is to procure a large number of such functional radios, okay, try to open them up. And if you open them up, you will end up finding that there is a complex circuitry which is behind it, giving it the property of making you listen to the songs or the news that is coming from a far off distance. And this electronic circuitry, which you don't understand, let's say we don't understand it very well, but we know that it is made up of a variety of components, which we know in retrospect to be resistors, capacitors, transistors, and other elements connected by wires. Now, a biologist would try hanging them on the wall, such a radio, and throw pebbles at them, stones at them towards the electronic circuitry. Something will be hit by the stones, and you will see that there is a change in function of the radio. The radio, which was otherwise functioning well, will all of a sudden, for example, would become mute, for example, or would end up giving static noise or something of that sort. This is the so-called loss of function assay that a typical biologist would tend to do. And then that part will be named by that loss of function. Something like RIC, the really important component, SRC, serious component, and somebody else will come up with another component, which is the most important component, MIC. And this hilarious story tells us about how we have ended up becoming experts in naming different parts of the cell by looking at loss of function assay. And somebody else will come up and say that, look, none of what you found is so important for this radio, but there is something called 𝐒𝐢𝐭𝐥𝐞, the undoubtedly most important component, which is the most critical element of the radio. This hilarious story was actually narrated for the first time by a Russian origin American scientist, Yuri Lazebnik, in this cancer cell article, which I strongly recommend all of you should read to be able to appreciate the core differences between a reductionist approach versus that of holistic approach. What Yuri suggested was that as opposed to looking at cartoonistic pictures of pathways and regulatory mechanisms in a cell, which is what has been shown on the top of this slide, we need to have a far better picture of how the cell functions, similar to that of an electronic circuitry that has been available to us for a while now. And then once you are able to put together such an electronic circuitry, you should be able to understand a living cell, a healthy cell, or for that matter, even diseases such as cancer, asthma, diabetes in a far better manner by virtue of putting the holistic picture at the center, which is required. And the take-home message where traditional biology lacks a formal and systems approach. And by the way, I must emphasize that for the last roughly 20 years, I would say that this approach is being taken increasingly by biologists, regardless of the fact that they are doing reductionist studies and assays. They would obviously think of putting it together to be able to understand it better as a system. And the second take-home message is that we need to have a holistic perspective for studying biological systems. That's what Yuri proposed way back, roughly 20 years back now, which is what I'm advocating through this hilarious story of a biologist who finds a radio. Notice, radio here is a metaphor for any complex system for that matter. And in the context of what we are discussing today in this talk, we are referring to biological entities. It could be a three-dimensional structure of a protein, or it could be cellular mechanisms which are underlying complex diseases. It could be an organ system such as heart, or it could be a brain-like organ system, which is created by virtue of large number of neurons interconnected in a subtle manner, giving rise to consciousness, cognition, and similar phenomena, which are emergent properties, I may say, right? So that's the story of uryl azabnik and a radio found by a biologist, and take-home messages that we have heard so far. So I believe that the central dogma can be possibly be extended to put the network or a systems perspective of any biological entity that we are investigating, whether it is protein structure or a disease for that matter, complex disease for that matter. When you think of biological entities and their graph theoretical or a network representation, turns out that a variety of biological systems at different scales, starting from nanometer scale, which are protein structures, to all the way up to macromolecular networks, such as protein-protein interaction networks, the gene regulatory networks, metabolic pathways, and a variety of omic interactomes, the transcriptomes and proteomes, etc. All of this can be seen from a network perspective. And not to forget, at a centimeter level, we have neuronal networks or functional networks of brain, which can be investigated from graph theoretical perspective. And finally, not the least, we can look at food webs, the ecological systems, which are operating at range of a few kilometers, again from graph theoretical paradigm. So let me take you into understanding these systems in a networks paradigm. And while doing so, I must also emphasize that while we can see a cell to be a protein-protein interactome or a metabolic pathways network or a gene regulatory network for that matter, in reality, all of these entities, all of these abstract representation of cell or cellular mechanisms are interconnected with each other, as shown in the picture here. A metabolite or a gene could be giving rise to a protein, and a protein further could be going and activating or deactivating a metabolic pathway in the metabolic picture. So all of these are very, very tightly integrated and interconnected with each other, and therefore creating a network of networks as we understand these biological systems. Well, that was my first part, to tell you about uryl azabnik's story about can a biologist fix a radio. The answer to this, way back in 2000 or so, 20 years back or so, was absolutely not, given the standard reductionist approach that biologists would tend to take until then. But in the last couple of decades, there has been extreme interest in application of systems biology, network biology, in the context of a variety of biological entities that we have been probing for a while now. Now, let me move on to the network biology and tell you about a bit about graph theory, the history of graph theory, and a variety of graph theoretical paradigms and parameters which are used for investigating any kind of network system for that matter. So I believe that many of you would not be acquainted with graph theory. And the agenda here is not to take you through the theorems and lemmas, but to give you an overview of what is this graph theoretical paradigm, where it all began, and how it can be, or any system for that matter, can be investigated from a graph theoretical perspective. The story began way back, a few centuries back for that matter, in a small town called Köningsberg, which has been depicted in front of you. And there was a river that was flowing through this middle of the town, and the river has been shown in the blue color, and there were 7 bridges which were located on this river. And the story goes, the anecdote goes in this manner, that the residents of this small town or a village, when they used to take a walk over these bridges, they would ask a question: is it possible for us to start from a given point, cross all the bridges once and only once, and reach back to the same place again? Would it be possible at all or not? Now, I would leave it to you as an open question to investigate that. The most obvious and intuitive way of looking at this question is to actually trace the path from one place to another place by going through bridges, the red patches, to see if it is possible to do so or not. But then that is where the genius of Euler came into the picture, who for the first ever time converted this problem into a graph. This is the same topology that I showed you in the previous slide, which he converted into a graph where essentially all contiguous land pieces that were shown here in capital A, B, C, and D letters were represented by vertices or nodes, as they are called, the red dots, and every bridge which has been shown using small letters, the small A, B, C, D, E, F, they were represented using a line between the two dots or the nodes. These are edges or links, as they are called. And then when you have such a graph theoretical representation, turns out that it's relatively straightforward to think about the logic behind the question that was asked and to see if it is at all possible to have a path as proposed by the town dwellers in Königsberg. And then the answer to this question is that no, it's categorically not possible in this particular topology that has been shown in front of you to actually have a path which is called as the Eulerian path, right? To be able to investigate graph theoretical basics, those of you who are interested in applications of graph theory or systems perspective in the context of biological entities, or even for that matter, non-biological entities, I strongly recommend looking at this book from a stalwart in this area called Network Science by Barabasi, which is freely available in an online mode, in an HTML mode. If you search for it, you will be able to get it in Google. Network Science by Albert Lazzaro Barabasi. Have a look at it. Now, I'm going to give you a very brief and quick primer of graph theory. What are the different elements of a graph? Nodes or vertices, and edges or links, as they are also called as. And these two entities together are good enough for representing any interconnected system which has got discrete elements connected to each other. And having got this representation of a graph which is represented as G and L, where N is the number of nodes and L is the number of links in it, turns out that systems such as computers interconnected with each other, or individuals who are interconnected with each other, for that matter, even proteins binding to each other, all of this could be represented using a simple graph as shown in the right bottom here. All of these entities could be represented in an abstract fashion as a graph. Now, it's not about the graphical or graph theoretical representation of systems which matters to us. What matters to us far more is that of what kind of questions we are asking, and we'll come to that in a bit. Now, turned out that beyond what Euler did way back in Königsberg, we need to look at systems today in 21st century, which are far more complex in terms of number of elements that are present in these entities. The number of elements is extremely large, and therefore computational intervention becomes extremely necessary. And that is where you need to find ways of representing a graph in a computational manner. And representations as shown in front of you, the adjacency matrix on the left hand side and something called edge list, which has been shown on the right hand side, are the ways by which a typical graph is represented in a computer. And we need to write algorithms for investigating, for analyzing these graph theoretical representation, the adjacency matrix and the edge list, or similar to that, to ask interesting questions on these graphs. So having represented any graph as a adjacency matrix, which essentially tells you whether node number 1 or node number I and J are connected by a link with each other or not. If there is a link between them, it will be put as a 1, entry as a 1. And if there is no link between the two nodes I and J, then it will be a 0. So it's a simple binary adjacency, symmetric, not symmetric, it's a binary matrix that we are having here. It may or may not be symmetric depending on whether the graph is undirected as shown here, or it is directed. As you can imagine, as a norm, a directed graph may or may not be symmetric depending on how the edges are impinging from one node to another node. So having got a representation in terms of adjacency matrix, one can find out a variety of parameters, or one can compute a variety of parameters of the graph. The easiest one, don't get bogged down by the mathematical equations which have been put in front of you. They have been put such that those of you who are more interested can actually look up later and try to understand them in a deeper fashion. But if you were to find out what is the number of links or edges that are connected to a node, if you want to compute that, then you can actually sum over the column or the row for that matter in the case of an undirected graph and find out what is called as a degree of a node. It's nothing but how many other nodes a given node is connected to. That's what degree represents. And in the directed formalism, in a directed graph, the same degree comes in two avatars. The in degree, how many edges are coming onto a given node, and how many of them are going out of it, the out degree of it. And all of this can be computed, as I said, by simply going through the sum over the rows or columns, depending on what you intend to compute in the case of a directed graph, right? So these formulae which are put in front of you corresponding to the computation which was marked in the adjacency matrix tell you a simple mathematical formula which can be used for computing the in degree, out degree, or for that matter, a degree in the case of undirected formalism. All right, that's what a degree is all about. So as you can see in the graph that has been shown on the left top, undirected graph, the degree of node A is 1 because it is connected to only one another node. And in a similar manner, degree of node B is 4 because it is connected to 4 other nodes. That's a simple notion that we have behind degree. And you have in degree and out degree as shown here. For example, the node C has got 1 out degree and 2 in degrees as shown by the arrows on the edges, right? So it's a very simple notion that we are looking at. And going further, one can do statistics to find out what is called as the average degree of the network itself by summing over all the degrees and taking an average of those. And similarly, you can have average in degree and average out degree that can be calculated. And some of these equations which are put in front of you are actually theorems which tell you how average degree can be computed once you know how many edges it is having L and how many nodes it is having, number of nodes N, right? And the same is the case in the case of directed graph, where you can compute by knowing the number of edges and number of nodes, the average degree of the graph. These graphs, undirected and directed, can be seen in the real life. For example, if you have a representation of co-authorship, who is co-authoring with whom in terms of research article co-authorship, as an undirected graph. Similarly, you have network of actors. Which actor has acted with other actor by virtue of shared movie can also be represented as an undirected graph. And in the biological context, a typical PPI, the protein-protein interaction network, also is an undirected graph. And in a similar fashion, you have directed graphs such as World Wide Web, the phone calls network, and the biological example of metabolic reactions in which a metabolite is getting converted into another by virtue of a metabolic reaction. And that's another example from biology of a directed graph. Going further, one computes degree distribution. This is simple statistics such as finding out the weight distribution or height distribution of individuals in a group. Exactly in a similar manner, knowing the degree of an individual node, you can find out what is its degree distribution. So on the y-axis, you have fraction of nodes which are having a degree 1 or 2 or 3 for that matter in this simple graph, right? As you can see that in the next graph, which is also called as a ring graph, all the nodes are having degree 2. They are connected to 2 other nodes adjacent to them, thereby getting a degree distribution which is very straightforward, kind of a delta function as it is called, right? So the reason why I wanted to look at degree, average degree and degree distribution is because there is an interesting property which has been observed in most real world networks, including biological networks, that their degree distribution tends to be having something called a scale-free nature. What is it that we are referring to? Unlike in a network, any given network, having same number of nodes and same number of edges as that of a real world graph, if it is randomly interconnected, if you put together an algorithm by which you can actually connect the nodes in an absolutely random fashion, then the degree distribution, the distribution of the degrees of nodes, will end up having something called a Poisson distribution, which is very, very similar to that of a Gaussian in certain limiting conditions. However, it looks as I just drew it for you. Unlike what a Poisson distribution would be indicating to us, the real world degree distribution tends to have the nature as shown with the dotted points, which is a scale-free degree distribution, indicating that there are a few nodes in this network which are having exceptionally large number of connections or large degrees, such as 80 and 90 in this simple graph of yeast protein interaction network. These are referred to as hubs. Having these very small number of exceptionally large, heavily connected nodes is what we are referring to as hubs. Similarly, I'm going to skip through some of these technical jargons, but giving you a glimpse of what it refers to. Something called clustering, which only talks about nothing but how well is a given node connected to its neighbors. If a node is connected to two of the nodes, then whether these two nodes are also interconnected with each other, well, that's the question which is answered by computing what is called as clustering coefficient, which ranges between 0 to 1. The higher the value, higher is the propensity or probability that neighbors of a given node are also connected with each other. As shown in the picture, clustering coefficient for red node I is the highest in this left-hand side picture, whereas it is lowest on the right-hand side picture, because none of the neighbors of I, the blue color nodes, are connected to each other, right? Having seen this, similarly, you have other parameters such as path length, how many paths or how many edges that you need to cross to be able to go from a node to other node, and correspondingly its average path length can be calculated for both the directed as well as undirected graphs as shown in the picture here, right? So you have degree, you have clustering coefficient, you have path lengths, and you have other properties which are computed using statistics of degree called assortativity or disassortativity, which is respectively for networks which are predominantly having nodes which are richly connected, heavily connected, which are tightly interlinked with each other, as shown here in the picture. On the contrary, you may have the heavily connected nodes connected to poor nodes predominantly, as shown on the right-hand side picture. So this is another property which can be measured using simple graph theoretical metrics. Why are we looking at all of these metrics and representing entities such as biological systems from a graph theoretical perspective? We are doing so because there are a variety of interesting phenomena which are in the purview of biology and epidemiology, such as spread of diseases, robustness of infrastructures, as well as modeling and control of biological entities, need understanding networks and their network properties. And that's what I'm going to tell you in the applications part. And there is a variety of questions that one can ask, such as how many elements that a graph has, how many interactions it is having, whether it is directed or undirected, is it weighted or unweighted, which I didn't touch upon, but you can imagine an edge being weighted or unweighted depending on the type of network that you are referring to. What is the nature of connectivity and connectivity distribution? Are there few nodes which are of special importance in the network for some reason? Is it important for the network to be cohesive, being together, or is it better for it to be spread along, spread across different subgraphs, as they are called, or components? What is the nature of degree-degree correlations? Is it assortative or disassortative? Are there special patterns of connectivity among the nodes which are important for performing specific function in the network, and how do you identify structural and functional modules? All of these questions can be asked in the context of all the network parameters and metrics that we can compute on these graphs. And we are referring to biological graphs predominantly in this talk. There is a notion of random graphs, which has been in existence for a long period of time. And mathematicians rather have been enamored by this idea of graphs or real world graphs being randomly connected with each other. Turns out that Paul Erdos and Alfred Renai were some of the couple of mathematicians who were behind growth of this random theory of networks, which I'm not getting into the details of, but you can actually create a random graph by taking a certain number of nodes and edges and thereby investigate its properties. And for the first ever time in the late 90s and in the early 21st century, it has been refuted that real world networks are actually randomly connected. Okay, and there are various ways by which random graphs can be created. There are a couple of models which have been in existence for a while, but then turns out that real world graphs are actually not random. They seem to be having certain preferential connections which are not part of random graph paradigm. And one of them refers to what is called as the small world networks. You must have heard this phrase that after all, it's a small world. This, in the context of network, means something very unique. Again, I'm going to go through these slides quickly and show you one of the landmark papers in the area of network science briefly, which referred to and for the first ever time highlighted the notion of small world network. In summary, without going into the details of this beautiful small research article published way back in 1998 by Watson-Stugaz, I'm going to tell you only one thing. That is that a network could be either completely regular, the way it has been shown on the left-hand side graph, wherein nodes are interconnected with each other in a very beautiful regular fashion, or it can be completely random as shown in the picture here on the right-hand side, wherein nodes are absolutely randomly connected with each other, or as Watson-Stugaz ended up finding that it can have a small world architecture, wherein there is a splash of regularity as well as a bit of randomness thrown in, which is what you end up finding in small world graphs. And all of these graphs, again, I'm skipping a couple of slides here for the sake of brevity, but then all these graphs tell us is that when you measure the clustering coefficient, which I mentioned about a couple of slides before, and its characteristic path length or average path length in a graph, turns out that there are three domains or three regimes that can be seen. One is that a graph could have very high clustering coefficient and a very high path length as shown in the red patch, which is what the regular graphs are known to be having. On the other side, they could be having very low clustering coefficient and low path length as shown in the yellow patch, which is what random networks are known to have. On the contrary, the small world networks have a combination of these two properties. They have very high clustering and a very low characteristic path length as shown in the green patch. So this intermediate zone, which is what is found by Watson-Stugaz, is what is critically important in the context of understanding real world graphs such as the C. elegance's brain network, which is one of the very well characterized networks, brain networks of neuronal architectures of living entities that we have. Or for that matter, even in the context of other networks, such as film actors connectivity and power grid networks in North America, they showed that all of these graphs are characterized with small world architecture. And those of you who would like to investigate or have fun with small world architecture, I would refer you to going to this Oracle of Bacon website and play with the challenge that has been given there in the context of film actors network. Well, having told you about the small world network, the last thing that I'm going to tell you before moving into the applications is the scale-free networks. Essentially, scale-free networks refers to the fact that unlike a random network which has got a Gaussian or a Poisson degree distribution, the real world networks tend to have what is called as a power law architecture or a power law degree distribution, suggesting that there are few nodes which are very heavily connected to other nodes. Okay, and this is what is referred to as the scale-free or power law architecture in the context of, or in the parlance of graph theory. And here is an example of road connectivity network, which has got a random connectivity, wherein most nodes are essentially connected to 3 or 4 other nodes or cities in this road architecture network. On the contrary, the airport connectivity network of North America has got a scale-free architecture wherein a few nodes are heavily connected, like Chicago or Los Angeles, by virtue of a large number of flights going from that, or number of other cities they are connected to by virtue of having the airline connectivity from there. All right. Now, I know that was a very quick and a rushing introduction of graph theory and its history. However, going through the book of Barabasi and slides, if you wish, which will be shared with you, my idea is that you will be getting a better understanding of the graph theoretical formalism which is used for understanding biological entities. Now, I'll quickly rush through and tell you about some of the applications in the next 5 minutes before closing, how graph theory has been used in the context of biology at different scales. The first example is that of the nano level architecture. The protein structures are nano machines, as we all know about, and these can be represented as a graph wherein each amino acid is a node and 2 amino acids can be said to be connected to each other by virtue of spatial proximity, they being close to each other in the 3-dimensional space. And when you have such graphs, they are referred to as the residue interaction graphs. In fact, this was topic of my PhD thesis, wherein we were among the early movers into this topic of modeling protein structure as a graph and trying to understand its structure, function, kinetics, and design part of it. And we incidentally ended up making an interesting observation and discovery of what is called as an assortative mixing or assortative connectivity in the protein structure, which is exceptional and is not found in any other kind of networks, including biological networks. Most of them are known to be disassortative, where rich nodes are connected to poor nodes. In this case, the rich connectivity is predominant. And going further, we could actually use these graph theoretical modeling paradigm to design a hyperthermostable enzyme, which has been patented by now, which has been found originally from a plant called Potentilla extrasanguinea, found in Himalayas. So that's the way you can model protein structure as a network to understand its structure, function, kinetics, and hopefully use it for designing proteins of whatever properties that you would like to have them. The next example is that of complex diseases such as cancer, asthma, and diabetes. The molecular mechanisms underlying these diseases can be represented as a graph, such as the one which has been shown here, and that is of cancer mechanisms, wherein each node is a protein, each dot is a protein, and a link, an edge between the two, refers to a physical interaction that has been empirically found to be present between the proteins. And in this particular paper, PLOS ONE paper that I'm pointing to, we investigated the properties of cancer gene network, trying to understand what are the mechanisms which make a cancer what it is. And is it possible for us to find out ways by which we can control the cancer by virtue of identification of potential therapeutic targets of cancer? And that's what we worked in this article. And again, I would like to point you to this research article for paucity of time. Further, and this research was done, previous research was done by my couple of PhD students, Vinay and Shikha. The other research that we have investigated and people have across the world have been investigating is what is referred to the term of controllability, which comes from electrical engineering, can be applied in the context of biological networks to understand which proteins are critical in driving the dynamics of networks underlying cancer, which is what was done by Vandana, my PhD student, who looked at the cancer mechanisms, the molecular mechanisms underlying cancer, and the controllability, as it is referred to in electrical engineering. The idea in these explorations of complex diseases using graph theoretical paradigm was also to find out the control mechanisms, the therapeutic targets. And that's what we did in this particular research article. This is work by another PhD student called Shivalika, where we looked at one of the important bottleneck enzymes in diabetes, the polyol pathway, aldose reductase, and went ahead to identify, using in silico drug discovery mechanisms, potential therapeutic target, therapeutic molecules, which can be, which were coming from serpentina, which can block the mechanisms of aldose reductase, thereby are therapeutic molecules against type 2 diabetes. That's what we did. And all of this research that we have done so far can be broadly put in this way, that by understanding disease and their therapeutic targets, and by compiling molecules from medicinal plants, which we worked on from mainly from Western Himalayas, and coming up with a virtual screening and molecular dynamics protocol, which can be used for identification of lead molecules. And that's what we worked on, which is being taken forward in an experimental direction. The therapeutic development pipeline or the drug discovery pipeline can be also potentially shortened by using tools from network biology in conjunction with in silico drug discovery, is what we showed by looking at the drug, target, and side effect networks. This is what is called as a tripartite network, wherein we investigated the data of drug bank and CIDR, these two databases which provide information of what proteins are interacting with which drugs from the cell, and what are the side effects that are known to be present for various drugs. Rakesh worked on this as part of his thesis, and we found ways using machine learning approaches and mathematical modeling for predicting the potential side effects of a given drug, which is what ends up becoming a bottleneck and increasing the time and efforts and money spent in the drug discovery pipeline. Towards the end, I would like to tell you that brain, which has been said to be one of the most complex organ systems that is present in the living entities, can be understood again from a network perspective, and has the availability of data corresponding to neuronal connectivity and functional connectivity among different parts of the brain, has promoted this kind of research heavily, wherein you can look at the networks in the brain by looking at the data coming from a variety of sources, including that of Human Connectome project, and similarly the functional connectivity of the brain networks, which can be investigated using fMRI data, which is becoming increasingly available today. And in one of the studies that we have done, Rahul worked on C. elegans brain network to understand the structural and functional connectivity, structural connectivity in the case of C. elegans and functional connectivity in the case of human brain connectivity in the context of schizophrenia is what we looked at. So I again wanted to give you a glimpse of a variety of systems that can be seen from a graph theoretical perspective in biology. And to end, I'm going to give you a glimpse of what we have done in the context of food, wherein we have created a new niche called computational gastronomy, where we have tried to see ingredients connected to each other by virtue of their shared flavor molecules, and ended up making an interesting discovery in the context of Indian cuisine. And we are now taking it forward in a variety of directions to, to come up with applications which can be used in the context of food. Well, that was a very quick summary in 40 to 45 minutes in understanding application of graph theory networks in the context of biological entities. I hope you've been able to get some take-home messages in terms of how one can use graphs and graph theory in the context of biological entities. with various interesting applications all the way up to drug discovery and understanding brain mechanisms. And I would share the slides with you and would again ask you to look at this book of Network Science by Barabasi to be able to deep dwell on it further. So thank you. Thanks a lot.
Thanks a lot, Dr. Bagler, for this amazing talk. So there are several questions in the chat box. So the way I'll do it, maybe I'll just read out the questions from the chat box so that everyone can also listen in, and then we can go through the questions one by one.
Sure.
I also request anyone who has any questions, please type in the chat box, and then we'll also take them. So the first question is by Ranjan Singh, and they are asking, sir, can you suggest any software for, I think, network analysis? Cytoscape, is it relevant to this or not?
Okay, right. So among the softwares, I would recommend a couple of them. One is the library, which is available as part of the Python programming suite called iGraph. NetworkX, which is part of Python, and iGraph, which is part of R suite. If you are interested or you are more keen to using the R programming language. Okay, so if you want to do interesting applications or utilization of algorithms that are already available and computation of network metrics, these two tools are extremely useful. Of course, those of you who are not oriented towards programming, then you may use Cytoscape in addition to the plugins that Cytoscape provides, especially for biological network analysis.
Right. So the next question is by Soham Karandikar. They're asking, in the graph theoretical representation of a protein, how are different kinds of chemical interactions distinguished from each other? So the interactions between two amino acids, how is it distinguished from each other? Right.
And I believe Soham is trying to ask here the context of different type of interaction. By that, I believe you mean, for example, hydrogen bonds, covalent bonds, non-covalent interactions, etc. That's what he is referring to. So the distance is taken as a proxy for representation of all kind of non-covalent interactions, Soham. The idea here is to bundle all non-covalent interactions together as an edge between the two amino acids, as long as they are at a distance of roughly between 7 to 10 angstrom. That accounts for all the non-covalent interactions which are responsible for the folding of the protein into its native state. And turns out that this is a very reasonable representation of protein structure, which can account for protein folding kinetics and also allow you to distinguish between different type of protein structures such as alpha, beta, alpha/beta protein structures. I hope that answers your question, Soham.
Right. So next question is by Soham. If anyone has any follow-up questions, they can also type in the chat box. So the next question is by Abhirupa. They're asking, can we apply this on microbe interactions?
Okay, that's an interesting question. In principle, yes. If you have, look at this, it's like there are a couple of things. One, do you have the data of representing microbe interaction as a network? If you have the data, of course, you can create a network out of it. The next thing which is extremely critical that you need to worry about, Abhirupa, is that having constructed such a network, assuming that you have that data, have some data which can be used for doing so, what are the kind of questions that you would like to ask on it? If network science and network parameters and network analysis, if it can help you answering those questions having constructed the network, then this is a meaningful approach to take. In the absence of having interesting questions which can be answered using graph theoretical paradigm, all of this is a futile exercise.
Next question is by Soma Sundaram. They're asking, what is the time complexity in finding clustering coefficient? Is it NP complete problem?
It is not an NP complete problem. Finding the clustering coefficient of a node and therefore that of a network is a relatively straightforward algorithm. If you wish, you can just shoot an email. I will send you a simple approach for finding clustering coefficient of it. It's not an NP complete problem at all. For that matter, finding shortest path length is a far more challenging problem compared to that of finding the clustering coefficient.
Right. Another question is, what are the applications of other graph centralities like betweenness and closeness?
Good question. So turns out that some of these parameters, such as betweenness and closeness, are very good in capturing nodes, especially betweenness, nodes which are critical for dividing the network into different clusters, different modules. Therefore, for example, in the context of, let's say, metabolic pathways network, if you would like to find out different pathway clusters or modules which are responsible for various biological functions, one could be responsible for amino acid synthesis, other could be responsible for, let's say, lipid synthesis. If you want to identify these biologically distinct modules, then something like betweenness comes into the picture. And for that matter, historically speaking, I can tell you in the context of what has been done so far, both degree and betweenness, in addition to closeness, have been used to identify the most critical nodes in a protein-protein interaction network or gene regulatory network. And by that one means targets. A typical target in a network which is representing a complex disease is identified using these parameters. And of course, there are other parameters like eigenvalue and similar kind of network parameters which are known to be of use depending on the kind of questions that you are asking.
Right. Then some people are asking, are there any free applications or softwares for performing this kind of analysis?
Right. In response to the previous question, all the tools that I mentioned, iGraph, R package, that is NetworkX, another library from Python, and Cytoscape are freely available. Cytoscape, you need to register as a academic user, if I understand it correctly now, but it's freely available.
Right. Tikshana is asking, Shweta is asking, for understanding network biology for beginners, what is the starting point?
Right. So my suggestion to you is to, first of all, understand network science, which is a very exciting topic in the first place, which talks about modeling both biological as well as non-biological systems as networks. And I would once again refer for the fourth time to the same book, which is Network Science by Barabasi, first to begin with. Beyond that, you also have books called Network Biology and Network Medicine, which are available. If you search for it, you will get it in Amazon. You can do it as a follow-up for dwelling on more biological systems as opposed to non-biological systems. That's the approach I would like to take, like you to take rather.
Then we have a comment by Sandra. She works with a European Commission, and she's saying that she's inspired with your gastronomy direction and what you see as a value of what has been achieved so far as a part of evolution of binary computing towards introduction of quantum computing to bring in the sensory science into the complex systems.
Okay.
So, yeah. Okay. Then I think. There's a general question by Prashanta Ghosh. They're asking, has anyone compiled what new developments in terms of novel drugs are being developed in India? And he's also commenting about the lack of new drugs that have been discovered in India and the need for discovering more drugs, indigenous drugs.
So I agree with the latter comment in terms of lack of drugs which have been discovered in India. I agree with that part. And for that matter, in general, new drugs discovered across the world. That has been on the lower side for a while now. Our research has been, unfortunately, I would say that's a part of the regret that I have, has been stuck with mainly doing the computational work and creating libraries of medicinal phytochemicals from the medicinal plants, and then trying to identify potential lead molecules. Beyond that, we have to collaborate with experimental researchers who do follow-up studies, including isothermal calorimetry studies, which look for binding of the protein structures with the small molecules, followed by in vivo studies wherein animal models are used. So unfortunately, we have not been able to take it further as much as we wish to, but we do have all the databases as well as research articles are anyway available freely. But in addition to that, the databases of small molecules are called Serpentina DB, Phytochemicals DB are again available from our side. Not to mention that there have been follow-up studies which have extended these databases in a humongous fashion. I don't recall the name of the database right now, but there is a database of phytochemicals which can be used as potential drug candidates from Institute of Mathematical Sciences, which has been there for a while now. And it is a superset of most of the databases that you see today in India. So you may want to go for that.
Right. And several people are asking for your email ID to contact you further. So we'll, if it's okay with you, we'll share your email ID with all the registered participants so that they can get in touch with you for any follow-up questions. Dr. Madhusudan Rao is the CEO of AIC, Atal Incubation Centre. So he's asking, given the poverty of data on interactome, is network analysis the next delusion?
Okay, well, that's a critical comment, Madhu. And I would say, in a way, it has been so that not having quality data of interactomes has been one of the limiting blocks in meaningfully using graph theoretical analysis on these interactomes in biology, right? So, but people have been enamored by the application of networks and graphs in the context of metabolomes, protein-protein interactions, etc. Oftentimes without worrying too much about, for example, the false positives that PPIs used to give earlier. Now, I believe that the techniques have been improved a lot to reduce the false positives, which was a serious problem in the case of PPIs. But you are right, when used in a manner that it doesn't inform us much, and when used in the starting with the basis of data which is not clean, definitely it's going to be a delusion. I agree with that part.
Right. Dr. Madhu, you have any? Okay, he's saying thanks, Ganesh. Yeah. If you want, you can unmute, and if you want to, you can ask also, sir. Okay, and then there is. So, do you want to say anything? Yeah.
Yeah, I just wanted to say, Ganesh, very nice lecture. Of course, I heard you earlier, and I keep reading your papers. But yeah, I always have that doubt. But thank you very much, and keep the good work.
Thank you. Thanks, Madhu.
Okay. So another question is by Tikshana. They're asking, is it possible to cross-check interactome results? What is the correct way?
All right. So it depends on what you would like to verify or cross-check. For example, if you would like to cross-check modules that are present in biological systems, just to give an illustration, E. coli metabolic pathways network has been very well characterized. Everybody knows about it. We know what are the different modules present in it in terms of different synthesis modules and metabolic modules that are there. These can be cross-verified by the topological modules or network-based modules which are predicted using any kind of module discovery algorithm. So there is a way to do that. In a similar manner, in any given system, if you know what are the important protein targets, just to give an example again, yeast protein-protein interaction network, one knows which are the lethal proteins. By that, I mean essential proteins, which when knocked out will lead to lethality in the organism. That can also be verified by looking at proteins which are having very high degree or high bitness. Do they match with them or not? So there are ways by which, so in each of these cases, you need to have empirical data which can be used to corroborate with what has been predicted by network analysis.
Right. Next question is by Ayushi. They're asking, sir, I wanted to ask if there are some genes which have not been annotated in the pathway databases, yet but sufficient information is available about them in the literature, then how should we validate their presence in our interactome?
So they are not annotated while enough data is available about the same. Well, I don't know how do we go about, sorry, I don't think I have an answer to your question. But I do know one thing for sure, that by using module detection algorithms and by finding which module does a given gene or a protein or a metabolite belongs to, one can try annotating the gene by virtue of for its functionality, is one of the things that I know. But I'm sorry if I'm not getting your question clearly. Therefore, I don't have a clear answer to you.
Okay. So maybe if they have any follow-up thing, they can get in touch with you. We'll share your email ID. So next question is by Somasundaram. They're asking how the community detection algorithms are useful in these applications.
Community detection algorithms have been of immense value when it comes to investigation of biological networks. I gave you one example wherein you would like to annotate a gene in the absence of annotations of being available for all the genes. Partially it is available. In that case, you can find out the modules, and if an unknown or rather a gene with unknown functionality, if it belongs to a module which is known to be having a clear functionality, then you can give a putative function to that particular gene. That is one of the possible applications of cluster identification.
We'll just take one last question by Tikshana, and they're asking if doing de novo interactome analysis, how can we cross-check?
Not possible, I would say. In the absence of having empirical data, I would say that only de novo analysis of a network wouldn't help you in doing any kind of cross-validation. You must have empirical data available, experimental data available to be able to come up with any kind of cross-verification. Without that, all of this is going to be wishy-washy.
Right.
Yeah.
So, okay. So with this, I think we'll stop here, but we'll share your email ID with all the registered participants, so that if anyone wants to get in touch with you for more follow-up questions or anything, they can get in touch with you. And with this, we'd like to thank you, Dr. Bagler, for conducting this amazing session, and to the participants for the amazing question answer session, and for joining in on a Saturday morning. So, and thanks everyone, and we'll keep continuing this webinar series every month. So hopefully we'll also see you next month. And please do also register for our newsletter, which is on which we'll release this month. And also do have a look at our India EMBO lecture series that we had initially shared, which will be happening October this year, for which the registrations are now open. So do check out the website, and also follow us on our social media for all the updates that we'll keep posting frequently. And thanks again, Dr. Bagler. Thanks everyone for joining in. So yeah, thanks a lot.
Oh, thanks a lot.